Hello, my name is Jennifer Jin. I'm a postdoc in Professor Yishi Jin's lab at UC San Diego. My research focuses on understanding how the brain remodels during development. Our brain is plastic and it can remodel its circuitry. And this allows us to learn and adapt to our environment. Our brain is most plastic during childhood. As adults, it becomes progressively much more difficult to learn a brand new skill like how to ballet, play a new instrument, or speak a new language. We can still learn, but it requires much more effort than children who can learn just like that. Now, what this tells us is that there's a window of time during early development when our brain is most plastic, which I'll refer as the developmental plasticity window. During this time window, our brain is remodeling its neural circuits. New connections are made or broken, and existing connections can be strengthened or weakened. This all still happens in adults too, which is why we can still learn. But the changes that happen aren't as long-lasting as in children. So things that we learn as adults often aren't as long-lasting as things that we learned as a child. Most of us can live without knowing how to play the violin or fluently speak 5 different languages. But imagine if we couldn't learn the basic skills like how to dress ourselves, if we couldn't read or write, or be able to tell when our family and friends are happy or sad. Intellectual and social learning disabilities are devastating and affect 1 to 2% of the people in the United States and around the world. But there's no cure. What is available is specialized teaching methods that rely on very early diagnosis, as early as possible, to target when the brain is still most plastic. And if they miss this period, then there is no cure. Imagine if we could shift or extend this developmental plasticity window to make adult brains more plastic again. Then adults with learning disabilities who missed very early diagnosis may have a better chance for effective treatment. And we might be able to also apply this knowledge to improve memory in Alzheimer's patients and also recovery after brain injury. A major factor that can move this time window is neuronal activity, the activity in our brains. But the field is currently at a roadblock, and we still have so many unanswered questions like, is the level of activity important? Is neuronal activity during this special period, is it different from the rest? And also the brain has so many different connections and also different forms of neuronal activity. Which ones are the important ones that make sure that our brain is plastic during early development? Well, wouldn't it be great if we could just look inside our brains to see what's going on? We can't do that in humans yet, but we can begin to study this using worms. C. elegans are microscopic roundworms, and their nervous system is much simpler than ours. But the fundamental properties are the same, like how neurons are connected and how they communicate with each other. And in fact, a lot of the important genes in neuronal function were first discovered in C. elegans. My research focuses on the motor circuit, which controls animals' muscles and movements. And this circuit undergoes dramatic remodeling during early development, and the entire process only lasts 5 hours because the animals only take 3 days to become adults. So what this means is that we can easily measure the entire span of developmental plasticity window using C. elegans. We also have advanced genetic tools and genome editing technology to visualize brain remodeling and brain activity. We can manipulate brain activity and study the mechanisms of this process at a molecular level. And also a huge advantage of C. elegans is that they're completely transparent, which means we can experiment on living animals throughout their normal development. And we don't have to dissect or take the brains out or potentially hurt the animals. My research goal is to find out how the activity in our brains contributes to restricting developmental plasticity windows specifically to early development. And using C. elegans allows me to bypass this current limitation of not being able to visualize what's going on in our brains. I'm using a sensor to directly measure activity in the motor neurons, and I found that the activity patterns are different before and after brain remodeling. To find the link between neuronal activity and brain remodeling, I'm testing different ion channels as sensors of neuronal activity, and I found potential candidates. I'm currently using CRISPR-Cas9 and other genome editing technology to generate tools to validate my observations. And it's very exciting because these ion channels also exist in humans. So my hope is that my research will contribute to discovering pharmacological targets to reopen this plasticity window later in life so adult brains can become more plastic again. Learning is a huge aspect of our development, and it fundamentally shapes who we are and our daily lives. If we can figure out what makes children's brains so much more plastic than ours, then this may open up incredible opportunities for adults and teenagers with intellectual and social learning disabilities, brain injury, or memory loss to learn the essential skills, gain the confidence to carry out their daily tasks in the hopes of improving their quality of life.